Hello there, my name is Faissal Sharif, and this is my presentation on the effect of psilocybin on neural oscillations and signal diversity in EEG and their psychological correlates. Psilocybin is a classic mind-altering psychedelic found in more than 200 mushroom species. After oral intake, its effects peak after around 90 minutes and last up to 5 hours. It binds to the serotonin 2A receptor, which is abundant in the posterior cingulate cortex, the PCC. Numerous studies have shown its therapeutic potential for depression, anxiety, and addiction, among others, which are thought to be mediated by the lasting increase in openness and well-being. Various EEG and MEG studies have shown a broad band reduction in spectral power under ayahuasca, psilocybin, LSD, and DMT. The most robust decrease in spectral power is found in the alpha band, between 8 and 13 Hz. Decreased spectral power has also been shown to correlate negatively with measures such as ego dissolution, intensity, and openness. According to the entropic Bain hypothesis, consciousness exists on a spectrum ranging from secondary states of suppressed entropy to primary states with high entropy. In the psychedelic state, during primary consciousness, our egos are dissolved, our thoughts are unconstrained, random, wishful, imaginative, and creative. Neurologically, this transition into the primary conscious state is reflected in the downregulation of the default mode network in the PCC. The impulsive complexity is a measure of the entropy that can distinguish between different states of consciousness. The increase of LZ onto psychedelics also correlates with subjective intensity of psychedelic experiences, ratings of well-being, ego disintegration, and openness. So, this study aimed to investigate the acute and long-term effects of psilocybin on psychological and neuronal outcomes and how those are associated. This study involved a one-arm, single-blind, fixed-order and Vivin subject design for which 28 healthy subjects naive to psychedelics were recruited. Beyond the acute measures administered during 2 dosing days, 4 weeks apart, long-term measures of psychological insight were taken during 6 time points up to 4 weeks after the high dose, the HD. During the dosing days, subjects received either 1 mg or 25 mg of orally administered psilocybin. This study compared the recordings at baseline and 2 hours post-dosing, equivalent to expected peak results. Moving on to results for psychological measures. For all 3 VAS, visual analog scale items, there was a significant main effect of time. Acute measures of degree of insight, richness of imagery, and strength of emotion were increased in the high dose compared to the low dose, and peaked 2 hours post-dosing. For the ego dissolution inventory, a dramatic increase of acute measures was found in the high dose compared to the low dose. For the psychological insight scale, there was again a significant main effect of time and significant increases between the high dose compared to the low dose. These stabilized between 2 and 4 weeks. Looking at Popo plots for each frequency bands, analysis revealed EEG spectral power decreased in the theta, alpha, beta band, and increased in the gamma band in the high dose compared to low dose. The most pronounced effects were observed in alpha. For entropy measures of LZ, significant, however, not as spatially widespread increases were found in the high dose compared to low dose. Moving on to the discussion. Changes in acute psychological measures support prior studies demonstrating the relaxation of prior hardwired beliefs and subsequent facilitation of psychological insight. Eyes closed richness of imagery ratings can be attributed to hallucinations and visual alterations commonly seen under psychedelics and dependent on 5-HT2A activation. Present findings of acute ego dissolution may underlie disrupted subjective processes of consciousness and the embodied self as seen elsewhere. And those aforementioned acute changes are thought to manifest in presented long-term increases of psychological insight, adding to present literature on lasting changes in personality under psychedelics. As expected, most pronounced and spatially widespread decreases were found in the alpha band and, to a lesser extent, beta and theta. We know that increased alpha is a signature for eye closure and relaxation, and that alpha is decreased when engaging with the world. The reduction of alpha seen here when disengaging with the world, meaning with eyes closed, therefore points to an altered sensory processing. Psychedelics bind to the 5-HT2A receptors, which are highly concentrated in the PCC and ACC, key areas of the default mode network. This binding, particularly in deep layer pyramidal neurons, leads to a decrease in lower frequency oscillations, particularly alpha power, and subsequent increase in intrinsic excitability within sensory networks, possibly explaining changes in consciousness and hallucinations. This shift from top-down to bottom-up processing is further discussed in the rebus model. The downregulation of the DMN, the seat of the ego, is likely causing subjective reports of ego dissolution and acute psychological insight herein, providing a possible link between the psychological and neuronal findings of the study. Since gamma 30 to 45 Hz overlaps with the broad spectral range of muscle activity between 20 to 300 Hz, observed increases must be observed with caution. Observed increases of Lamp-Ziff entropy replicated findings of previous studies on psilocybin, LSD, ayahuasca, and DMT. The findings are consistent with the entropic brain hypothesis introduced earlier, or according to which the richness of conscious states can be indexed through measures of entropy. With increased Lamp-Ziff as a measure of entropy, this study further substantiated the notion that psychedelics alter states of consciousness through the increased spontaneous brain activity, allowing acute and lasting changes in mood and personality. These changes are likely mediated by 5-HT2A dependent enhanced neuroplasticity and associative learning under psychedelics. Thank you very much for your attention, and these are my acknowledgements. Goodbye.